Hello and welcome to the next episode of the Psychedelic Medicine Podcast. I'm Dr. Lynn Marie Morsky, your guide on this journey, and today we're going to be discussing whether your genes can predict response to MDMA therapy with Dr. David Rabin. Dr. Rabin is a board-certified psychiatrist and neuroscientist and the co-founder and chief medical officer at Apollo Neuroscience. In addition to his clinical psychiatry practice, Dr. Rabin is also the co-founder and executive director of the Board of Medicine and a psychedelic clinical researcher currently evaluating the mechanism of psychedelic-assisted psychotherapy in treatment-resistant mental illness. Now, before we get to Dr. Rabin, just a reminder that the Psychedelic Medicine Podcast is for educational and informational purposes only. Nothing here is to be construed as medical or legal advice. And one last reminder, if you are a clinician and you would like to feel more comfortable discussing psychedelics with your patients, please check out the Psychedelic Medicine Association. We are dedicated to get you educated and up to speed on psychedelics, so when you have a patient who comes in with something psychedelics may be able to address, you will feel confident to have that discussion with the patient and perhaps refer to a psychedelic-assisted therapist when appropriate. For more information, please check us out at psychedelicmedicineassociation.org. Now, without further ado, thanks so much for joining us today, Dave.
Thanks for having me, Lynn Marie. It's a pleasure to be here with you.
I'm really excited about this discussion because this is something we have not discussed previously. I think way back in the day, there had been some white paper hypothesizing on things like, can we do genetic tests that might tell us who is going to respond to things? But I have not seen such in-depth research as the piece that you and your team put out. And now for everybody, I'm just going to read the title. We will put links in the show notes. And just so you know, Dr. Rabin is going to break this down in layman's terms for us because I'll read the title. You're going to be like, okay, next podcast. This is too complicated. I promise he will explain it to me in ways that I can understand it, and you will be able to understand it too. So the paper is called Pilot Study Suggests DNA Methylation of the Glucocorticoid Receptor Gene, NR3C1, is associated with MDMA-assisted therapy treatment response for severe PTSD. So, Dave, if you could start by telling us a little bit about what led you and your team to go down this line of investigation.
For sure. And again, thank you so much for having me. This is one of my favorite areas to talk about because it's literally on the forefront of mental health research. And there's been nothing like it out there to date in terms of the effect that we're seeing from psychedelic-assisted psychotherapy delivered properly. Key caveat there, right? And for a long time, I've been, probably now the better part of 20 years, I've been studying chronic stress and trauma and the impact of trauma on the body and the mind, and how it can prevent us from recovery from illness when it's not addressed. And we see this in veterans, in particular with severe PTSD, women who have been, victims of abuse, et cetera. And really, the folks who this impacts the most are vulnerable populations. They're people who don't have good access to care, and they're people who don't have a lot of resources to pay for good quality care. And so they're just kind of at the whim of the medical system to decide, well, here's what's available to you at a price you can afford. And most of those treatments, unfortunately, just to catch everybody up, most of the treatments that we have available today for PTSD that are the FDA-cleared considered gold standard treatments, only actually induce remission from illness. So remission meaning that you're no longer meeting diagnostic criteria for that illness, PTSD in this case, in about 30% of people. So of the 100% of the millions of people every year that are getting treated for PTSD with the best treatments that we have that are currently available, only 30% at best are getting better long-term. Everyone else has a diagnosis of PTSD for life. They have a much higher suicide rate and hospital admission rate than everyone else. They have a much higher healthcare cost than everybody else. And they wind up usually prescribed prescription medication that has pretty significant side effects for their entire lives to manage their symptoms. And they are still sick. So that's kind of where we're at today. And the reason why I became so interested in the research study, in the research around how psychedelics work, is because when you look at the current studies of psilocybin therapy for depression, MDMA for PTSD, And then you go back in time to the ancient tribal traditions around ayahuasca and why ayahuasca is used in tribal culture, how they use it, why they use it, what they think it does. Everything comes back to trauma. And that there is this idea in both Western and ancient cultures that trauma that's unresolved, unprocessed, that just lingers in our bodies and our brains for an extended period of time prevents us from healing and from achieving our full potential as human beings. And it leads us to feel unsafe all the time. And so this is what we were seeing in our veterans and our patients with severe PTSD that weren't getting better in the general population. And I went back to the literature and saw that most of my colleagues who had studied these folks were seeing the same exact stuff that we're seeing. People are just not getting better. So I started to explore other techniques like MDMA-assisted therapy, psilocybin, ayahuasca, ketamine, anything under the sun that could help, that might have evidence. And what I found starting back in 2012, when I started to enter into this area, was that there were substantial studies. There were at least 10 studies published in fantastic scientific journals, even back. So I was trying to find anything that could help for these patients. And I started to look at literature starting back in 2012 around psychedelic-assisted therapy, including MDMA, ketamine, psilocybin, and just anything that had been published. And what became very clear, even that far back, 12 years ago, was that people who had treatment-resistant depression and PTSD were having long-term, not only much better responses with just 1 or 3 doses of medicine, but of psychedelic medicine with therapy, but they were having long-term gains that lasted beyond the medicine administration. So after the medicine was stopped, after the therapy was stopped, people were still sustaining remission or even getting better. And this is actually what the Nature Medicine publications of the MAPS phase 2 and phase 3 trial have shown, which are really astounding, which is that when you take people with, on average, 17.6 years of non-responsive PTSD that's never gotten better, and you give them 3 doses of MDMA plus 42 hours of psychotherapy delivered over 12 weeks, we see that there's over 50% response rates, meaning, and closer to 70%, I think, of people actually experience some benefit, but 55% or so of people are no longer meeting diagnostic criteria for PTSD after the medicine sessions have ended. And then you look 1 year out, how are these people still doing? That number goes up from 55% to 67%. So to give you an idea of how impactful that is for the field of psychiatry, we have never seen in the history of our field any treatments that continue to work after the treatment is over. And we've never seen any treatments that have this kind of response rate for people who are this treatment resistant. So this is akin to what we're seeing in terms of the results with MDMA, just to sum it up, because that's what's been published the most on to date in top tier journals like Nature Medicine in very, very rigorous scientific trials, is that MDMA assisted therapy for people with PTSD is having an effect that's similar to what we saw with the discovery of antibiotics for infection. It took antibiotics discovered for infection by Fleming, in the 20s or 30s, transformed infection, which was a terminal illness that could result in death or amputation or long-term, lifelong issues, to a treatable illness that was actually curable within weeks to months. We're now seeing the same thing with MDMA-assisted therapy. So that is the background framework of kind of how I got here and why I'm so passionate about studying this area. So the thing that seems to be most messed up in people with PTSD is their stress response. And the stress response, again, is governed in large part by the cortisol receptor and cortisol system. And so, but it was never known until Rachel's work that there were actually changes to the cortisol receptor structure and function as a result of trauma. And so Rachel showed that there was this correlation between people with severe PTSD, like Holocaust survivors and their offspring, and these changes to cortisol receptor function that looked like if you were traumatized, you could actually pass those changes down to your offspring. And this was a really, really big deal, like a huge finding. It was one of those huge findings that was so significant that no one believed her when she published it. And because it was so far afield from the way we were thinking about PTSD back 30, 40 years ago. which is a good sign of any amazing discovery. If you look at most Nobel Prize discoveries, most of them have not been believed when they first were published. So fast forward to today, now tons of people have replicated this in causal animal models where they've traumatized mice or rats and then watched the cortisol receptor change, and then seen that uh, when you extinguish the learned fear, meaning you recalibrate the animal to safe environments, that you can repair or watch the repair of the cortisol receptor. And so this has been known. This has been known for a very long time. And the reason why this is so interesting, the second reason why we looked at this, is because Eric Kandel won the Nobel Prize in 2000 for discovering how we learn and remember things. And he hinted at back then that epigenetic changes had to occur to create new proteins, and that new proteins are which are in large part building blocks of our cells and how they function, cortisol receptors being a protein and cortisol being a protein hormone, that when these are made, that that we can that we actually need them to create that sustained change that lasts over a long time. So if we learn something right now and we don't have the epigenetic changes in the cells, then our neurons don't grow in the directions they need to grow to build new connections, right? So we have to have a change that is long lasting. And there's very few things in the body that last for months or years, like that would be responsible for the outcome that we're seeing in these MDMA trials, right? People at 1 year out from treatment are better off than people who were just finishing treatment. Something has to be sticking around that's responsible. So between those two things, we started to look at the cortisol receptor. And that was where we said, okay, we need to understand what's happening, if anything, to this receptor. And in this receptor system, is MDMA-assisted therapy starting to induce a repair at that receptor site, because that receptor site is damaged, for lack of a better word, by trauma. So if that receptor functioning and structure is damaged by trauma, then can we see repair with MDMA when people are actually getting better? And will that repair on that receptor site actually be proportionate or correlate with how much better people get? So if people get much better from MDMA-assisted therapy, their PTSD goes way away, do we see more remodeling at the receptor, or the opposite? And what we ultimately showed in this publication that was really groundbreaking and exciting is that, yes, in fact, the hypothesis is true that folks with severe PTSD who went through this trial, when they received MDMA-assisted therapy, they saw a statistically significant change in terms of repair of the cortisol receptors epigenetic code, which reflects improved structure and function of that receptor site. But not only that, we saw that the amount people got better, to the original title of the show, the amount people got better was directly correlated with, in the same direction as, the amount of remodeling and repair at the receptor site. So now there's a linkage between a biological target on the genes that we can look at through something as simple as a saliva in a spit cup. And we can look at that before and at different time points in your treatment and understand in the future, we're not here yet, but we can start to understand, are you responding not just based on your symptoms, which are self-reported, but are you responding based on the shift in your biology? Right. And your stress biology being the fundamental thing that's disrupted in PTSD, to bring it full circle.
Yeah, that's incredible. I have so many questions. First off, since you gathered saliva before they took the MDMA,
Right.
and were you noticing this, like she had found back in the day, that the changes to the glucocorticoid receptor, were they pretty uniform amongst the participants? Like, did they all have, because they were all severe PTSD patients, correct? And so in theory, they would all have kind of similar looking receptors.
Yeah, I mean, those studies have already been done, so we were not focused on
Okay.
replicating that. We knew that there were certain receptor sites like NR3C1 that are particularly responsive to trauma, meaning that there's more changes at certain sites in response to traumatic events that are unprocessed that result in a diagnosis of PTSD. So when you've had not just any trauma, but when you've had PTSD that lasts for an extended period of time, that there is a change in this NR3C1 site. Chances are there's actually changes occurring throughout the body and throughout the epigenome, but this site is one of the most sensitive sites to predict trauma. So we did see and we didn't look at the whole MDMA therapy trial sample because not everybody who was in that trial consented to participate, but we looked at as many people as we could. And ultimately what we saw was that there was statistically significant methylation at that NR3C1 cortisol receptor site, which is consistent with having a diagnosis of PTSD from past studies. And that after MDMA-assisted therapy, when the 3 doses, 12 weeks, when they spit in the cup again, that we start to see repair, significant repair.
So to put it into layman's terms, methylation is the kind of damage done by PTSD, and you were seeing a reverse of the methylation after the MDMA?
Exactly.
Okay, very, very interesting. And because I think we're all trying to envision this precision medicine type future where things like that, like you said, it's not tomorrow, but in the future, it would be nice to have these quantifiable, because maybe it's somebody is their demethylation is improving, but you can see that they're not at what looked like the endpoint that maybe the people in remission would get to. And so perhaps that'd be an indication for more treatments, or what might that look like?
Yeah, exactly. Right. So again, more studies need to be done. This is the first study of its kind. It's a pilot study. So it's really just throwing it on the field that, hey, this is something we need to be looking at, right? Because a big reason why insurance, going back to what we were talking about at the beginning, why insurance is hesitant to cover this up front is because they are not certain who's going to be in the 88% of responders from the phase 3 trial. We saw an 88% response rate. So who's going to be in the 88% of responders and who's going to be in the 12% that didn't respond, right? And they don't want to pay for the 12% that don't respond. So what we need to do based on this pilot data and what we are doing as our group, but what other groups are starting to do now as well, and anybody who's interested, who's listening, who might be interested in working together with us, we are happy to work together on this because it's an enormous endeavor. But basically, we're collecting larger sample sets from patients who are going through ketamine treatment for depression and PTSD. We're collecting saliva samples for people going through ayahuasca in the indigenous tribal setting. We're going to be collecting saliva samples from recreational users of psychedelics without therapy. And then psilocybin as well, because if we can get more data around what people's genetic code around or epigenetic code around the cortisol receptor looks like before and after, and at different time points, then we can understand what is the saliva signature of healing, right? Based on your saliva that you're putting in this cup at these different time points of your before, during treatment, and after, how close are you to getting to a signature of your cortisol receptor that looks like it's significantly repaired to result in the long-term gains, right? Because if people just relapse again and they have to go through the treatment again, then we're not necessarily solving the problem. In fact, we're not solving the problem. So the goal is, how do we get from zero benefit to these patients where they're suffering every day with this diagnosis to remission at the end of the study with highest likelihood and long-term gains? So the way that you might envision this in 5 to 10 years when we have more data is this will be precision medicine, similar to the way we use antibiotics right now. So this could be when you come in with an infection or a potential infection, they culture the infection, they figure out what is the antibiotic, they figure out what it's resistant to, and then they prescribe you an antibiotic that bacterial infection is going to respond to with a very high likelihood. And they give you, here's your prognosis, right? 1 to 8 weeks of treatment with X will result in this likelihood of you getting better. And we don't have that for mental health right now. So what this does for mental health in the 5 to 10 year time frame is it brings objective biological precision to our decision-making process that we've never had before. So if we can understand the cortisol receptor's signature, meaning what does your cortisol receptor look like when you're healed versus when you're symptomatic with PTSD, and then what it looks like in between along the healing process at different stages, then you can come in, you'll be able to come into the office in 5 to 10 years. I'll as your psychiatrist, I'll do your history and the same regular workup that I do, but I'll have you spit into a cup at the end of your visit. Then we'll send that for epigenetic analysis, and we'll be able to understand from your subjective reports and your intake and from your DNA what you need to heal. What is the path to remission? Not you're likely to be sick for life. Sorry, take these SSRIs forever. It's going to be, based on the study data, you have an 88% chance of responding to MDMA-assisted therapy based on your diagnosis of PTSD and your epigenetics code of your cortisol receptor coming in. So that's the first one that we're going to use for you. We estimate that within 12 weeks with 3 doses and this many hours of therapy, that you have a 75% chance of no longer experiencing PTSD symptoms anymore, right? You have a 75% chance of entering remission. So that's a lot more like the approach of what we're doing with antibiotics. And then we'll also be able to help decide over time, as we have more studies with different medicines, whether somebody is a better initial candidate for ketamine or for MDMA or for psilocybin or for any other substance, or even for therapy, right? Maybe there's somebody who's a better candidate for CBT than for MDMA with psychotherapy, right? If that's the case, we should be using that. We should be using the least risky intervention first and then escalating up. Ketamine is lower risk than MDMA. Maybe we should start there for most people, but we have to get that data. So that's how this will be transforming the field of mental health in the next 5 to 10 years.
I'm so excited for that future. That's fantastic. And because I have already failed the listeners at this point, I need to go back and ask you, would you mind explaining what epigenetics are compared to just regular genetics? Because you keep referring to like the epigenes of the cortisol receptor.
Yeah, absolutely. So sorry, I didn't mention this earlier. So when I talk about gene expression, meaning what does our DNA turn into, that's regulated by something called epigenetics, which means on the DNA. So if you think about what genetics means, genetics really means in the DNA. And every single cell of our bodies, which is quite miraculous, every single cell of our bodies except our sperm and egg cells have 100% the same DNA. They have the same base code. But a skin cell somehow knows to be skin and a brain cell somehow knows to be brain, despite all having the same instructions. How is that possible, right? And this is one of the miracles of biology and nature that's been discovered over the last 50 years, which is that the reason why our skin cells know to be skin and our brain cells know to be brain, even though the DNA background code is identical, is because there's another layer of code on top of the DNA called epigenetic code. And the epigenetic code, meaning on the DNA, tells certain parts of the DNA to turn down and other parts to turn up. And so in skin, our skin cells have all of these epigenetic code features that say, hey, turn down brain, turn down gut, turn down sperm epigenetics because you're skin, right? And then we're going to turn up all the skin stuff. And then in brain, it says, turn up brain, turn down skin, turn down gut, turn down sperm epigenetics because we're brain, right? And so it's really just another layer of code that helps determine what our cells do based on all of the DNA that we have and which parts of it are being expressed, going back to this term gene expression. We don't have 100% of our DNA expressed in every cell. We have a giant base code that codes for our entire human being, and then only certain parts of it are expressed in each cell. So where this becomes new is that we did not necessarily know that epigenetic code could be modified by experience from life, right? We knew that epigenetic code regulated DNA and helped our cells determine what to turn up and what to turn down, but we didn't know until Rachel's work that epigenetic code is actually something that can be modified by experiences in our environment. And trauma or PTSD diagnoses, series of significant traumas over time, can in fact change epigenetic code of the cortisol receptor gene. And that changes how the cortisol receptor gene gets made, and then it changes its function. And so that is kind of the stepping stones between our base code, our epigenetic code that regulates what is turned up and down, and then how stress and trauma actually shift the way that our code is expressed, particularly in stress response genes. And lots of genes probably, but what we know most about is stress response genes. So the best way to think about this, according to Eric Kandel's work around Nobel Prize winning work around memory formation and storage, is that epigenetics is basically like a memory. So your skin has an epigenetic memory that it's skin. Your brain cells have an epigenetic memory that they're brain. And when we experience trauma and that trauma is not resolved and we wind up with a diagnosis of PTSD, our cortisol receptors store the memory of that trauma and that stress. And so that is effectively what epigenetics are and how they shape our entire body's functioning and based on experience.
Thank you so much for that explanation. Much clearer picture of what's going on now. And the other question I had was, are you noticing that the, let's say, healed state, the patients that are having more successful outcomes, is it that they're having a larger change from their original like methylation state to afterward? Or I guess my question is healing more based on the difference between what they started off as, or is there kind of an absolute look to the epigenetics in those who have arrived at remission?
So we don't know that yet because we haven't done studies of that large scale. We are working with some folks who are doing that work now, but it's going to take some time. There's likely to be tons of stuff changing as a result of chronic stress and trauma and PTSD and mental illness, not just this one cortisol receptor gene. But to answer your question simply, what we know now definitively is people with PTSD or a history of severe trauma have methylation changes to their cortisol receptor epigenetics that change or impair the cortisol receptor's functioning and structure. When you experience healing, and we can define an example of healing being MDMA-assisted therapy because we see the healing response in PTSD. We see over 50% of people getting better. We see over 50% of people long-term no longer meeting remission or no longer meeting diagnostic criteria because they've entered remission. That when you look at those people before and after they receive treatment, that there is a statistically significant remodeling and repair at that cortisol receptor site, which is consistent with what your cortisol receptor looks like when it's functioning at its best. We don't have the massive data set required to understand, are we getting better than best, or where are we getting to, right? We do know is we're starting to initiate through the healing process of PTSD with MDMA-assisted therapy, a significant repair at this site. So there is actually, again, also interesting, a mind-body connection that's happening, right? You feel better mentally, emotionally, you subjectively report that you're no longer having the same kinds of symptoms, and your body's recording it in the DNA that is not just in your brain, it's in your saliva, right? So it's probably cortisol receptors that are impacted throughout your whole body. And so there's a whole lot of studies that are coming out of this, but that is kind of the gist, is the repair, and that there is a statistically significant improvement in cortisol, apparent cortisol receptor functioning before and after people receive these healing treatments in the context of PTSD.
That's incredible. I'm so excited for one, that you've already done this research, but all the research that's to come. Is there anything that you or anybody else is recruiting for, or like you already put out the call, like if any scientists out there want to help us with this, but we, for our audience, is there anything that you would like people to be part of where they could contribute to this?
Yeah, so I think, on the surface, the first thing is that, it is going to require studies of thousands of people over the next couple years, the next 5-10 years, to start to get the answers to these very important questions you're asking, right? These are the questions everyone wants to know the answers to. And answering these questions is going to shift the entire trajectory of the way we treat mental illness, probably fairly quickly. Like 5-10 years for these kinds of things is a relatively short time. Ideally, we'd have it yesterday, but it's going to be 5-10 years, right? So we need scientists and researchers who are interested in this area, particularly those who work in psychedelic spaces, to jump on board with us and even just do the same thing we did with MAPS, where they say, hey, we're running a study of psilocybin therapy. Can you send us sample collection kits so that we can collect data before and after? That's all we need. We'll even provide the sample collection kit. So what this is basically zero expense to the folks running these studies. So call to action, first one is if you're running a study of psychedelics in humans, please reach out to us. We'd be happy to help talk about how we can help collect some of the saliva data in your study by adding on a protocol that doesn't impact your protocol or study at all in any kind of negative way. It actually adds a lot of positive to it because we can show a biological improvement that supports the research findings. So that's number one. So please reach out if you would like to collaborate with us. We're a very fun group to work with. We partner. This is an initiative that's really coming out of Dr. Candace Lewis's lab at ASU, Arizona State University. So this is a very exciting opportunity to be able to collaborate and figure out how these medicines are working. The second participation opportunity that's really exciting is that we developed a technology based on this understanding of how safety and stress response and fear are fundamental to people struggling with PTSD. We kept pulling on those threads, and we were able to develop a technology called Apollo that delivers soothing vibrations to the body that activate the safety response system in the body on the go. So that is really interesting because PTSD is a disorder of learned fear. How do we treat PTSD? In animals and humans, we provide the same stimulation that they were getting that they are now afraid of in the context of safety. That's how exposure therapy works. That's how MDMA therapy works to some extent. All of these tools and techniques work to treat PTSD effectively, even though some of them are hard and don't have great response rates because the effort is high. They work in the same way. It's called safety conditioning. PTSD is a disorder of fear learning. So how do you fix fear learning? You learn safety again, right? So it's really that simple. And so Apollo is a wearable tool that allows people to get access to those safety states literally on demand, on the go. You can just have a little pod that you wear anywhere in your body. You tap two buttons, all of a sudden your body is feeling like it's getting a hug. And that increases vagal tone, which is the activity in our vagus nerve, is the primary nerve responsible for recovery and healing and safety and putting resources back into those parts of our bodies. And there's lots of vagal nerve stimulators out there, but most of them require you to take time out of your day to use, and you can only use them about 20 minutes a day. So we developed one you can wear all the time that has no side effects. And it's actually full circle, the first technology that's being used in psychedelic therapy to augment psychedelic therapy. So the reason why I bring this up is because the Apollo is the only wearable technology and the only technology that's actually being integrated into the MAP studies to sustain remission from PTSD long term. It's being used in thousands of patients getting ketamine treatment every year. And it's being used in clinical trials of psilocybin and ketamine for first responders. There's no other technology that's being used to do this. And the reason why is because if we can improve people's experiences with psychedelics by using a non-invasive, sound-based wearable technology without side effects, then that's better for everyone because the outcomes go up. And so the challenge with that study is that, like any study, recruitment is hard. And so the study we're recruiting for right now, that's a top priority for anyone who's listening, is that if anyone listening has been a participant of the MAPS MDMA-assisted therapy trials or knows anybody who has been a participant, if you're a participant, you're eligible to receive a free Apollo wearable, which is $349 just for participating in the study and helping us gather the long-term 2 and 3 year outcomes data following MDMA, because MAPS stops collecting data at 1 year. So we are the protocol that's collecting 2 and 3 year outcome data with and without Apollo. And so anyone is able to participate if you have been a participant in the MAPS MDMA trials, and you can get a wearable for free. And we are happy to get you set up and connected. And you joining the study is a critical part of us helping to understand even further how critical the safety response system is and how much better benefit we can get by augmenting that system. So for anybody who's listening, please reach out. You can find access to the study screener on the Apollo Neuro website under the research tab. And you can get access to a free device for your recovery.
Amazing. And where can the people from Call to Action number 1 reach out if they want to, on the scientist end?
Yeah, call to action number one. If you're a scientist studying psychedelics and you're interested in figuring out how they work and being part of our epigenetics cohort, we are happy to chat with you. Just reach out to me directly via email. And you can do that. Probably the easiest way is just to go to my website, which is drdave.io, and you can reach out there and through their email contact. Or worst case, if for some reason you don't hear back from me, just reach out to me on socials at Twitter and Instagram at Dr. David Rabin.
Perfect. Thank you so much. And thank you for the research you all have done and for coming on and explaining it to us. I look forward to having you back on when we have more very cool precision medicine things to share from your upcoming studies.
Sounds great. Thank you so much for having me.
Absolutely. All right. For everybody else out there, until next time.